Hello, everyone. Welcome to our series on using proficiency testing data to improve the performance of your molecular laboratory. My name is Ian Hagemann. I'm speaking with you from Washington University School of Medicine in St. Louis, Missouri. This session focuses on lessons that can be learned from multi-analyte next-generation sequencing proficiency tests. So in the talk today, I would like to tell you a little bit about how proficiency testing exercises can be designed to look at next generation sequencing testing and some of the lessons that can be drawn from those. The bottom line for this session is that next generation sequencing is the essential technology for testing multiple genes on a single platform. And of course, as part of offering high quality clinical testing, we need to make sure that our tests are validated. It's not possible to validate every possible variant, and it's also not possible for proficiency testing vendors to offer challenges for every possible variant. So I would like to introduce the idea of representative validation and methods-based proficiency testing, and explain how those approaches can help to ensure the quality of testing. So in contrast to single gene approaches in which a single assay looks at a single variant or a single gene, usually one gene per tube, it's now very common to perform panel testing using next generation sequencing for looking at multiple targets in the same assay. Often these will be assays designed to look for actionable mutations in cancer, and so will encompass multiple targets in a single assay or run, and potentially we can detect multiple different variant types in multiple genes, potentially single nucleotide variants, multi-nucleotide variants, structural variants, copy number variation, insertions or deletions. So our ability to detect all of those variant types does need to be validated and subjected to proficiency testing. Here's my overview of NGS-based testing. We start from nucleic acid. We prepare a library. Usually there's some form of enrichment, either by hybrid capture, as is shown here, or by an amplicon type of method to give an enriched library. And we subject that to sequencing, and then we align the short reads in step 7. Then we carry out informatic processing in step 8, first to identify the presence of variants, and then to annotate those variants using various databases, and then finally signing out a clinical report, which goes into the electronic medical record. So that's the basic workflow for NGS-based testing. Here's a little bit more concrete example of an NGS-based assay. This is only a typical example. So these panels might include a number of genes, maybe 41 different genes relevant to cancer. We'll sequence all exons of these genes. We might also sequence selected introns in order to be able to identify translocations. And the test design includes a specification of which exact types of variants can be called. And we'll always have pre-specified what type of specimens can be accepted, and based on our validation, we will have a limit of detection specified for the assay. So how could we validate these types of assays? We can't possibly test positives and negatives for every single type of variant or every variant that the assay can theoretically detect. But we can carry out representative validation, validating selected genes as a surrogate for the whole panel. We need to make sure that our validation set includes a sufficient diversity to put the test through its paces. So we need to be able to include within our validation set every different type of variant that we plan to detect. And we need to test the performance of our assay at a range of variant allele frequencies as well. And if the validation set has sufficient diversity, we can view that as a surrogate for the whole panel and thereby validate our test. The mirror image, or the flip side of representative validation, is what we've come to call methods-based proficiency testing. As viewers will know, it's important to participate in proficiency testing within a CLIA certified testing environment. And so laboratories will either subscribe to a proficiency testing challenge or engage in some type of alternative assessment. Those PT surveys or subscriptions or exercises should challenge the test to make sure that it's functioning according to its design specifications. It's not possible to challenge every possible variant or every possible gene for a couple of reasons. One, there's too many possible variants in the reportable range of these NGS assays. And the other is that in general, every lab has a slightly different assay. So it would be very difficult to design a proficiency testing survey in which every lab was able to detect every variant because the gene list may be different. So one approach that the College of American Pathologists has taken is to send out proficiency testing surveys, such as this NGS solid tumor mutation panel, that include a variety of variants. So these are engineered samples containing variants in a variety of genes, recognizing that subscribers to this survey don't necessarily test every gene in the survey. So one of the questions that we ask is, does your lab test for this variant? Out of 111 participants, there was really only one variant that every lab even said that its panel was looking at. That was KRAS codon 13 among the variants that were included in this particular mailing of this particular survey. The other thing I wanted to highlight is that when we look at lab performance, performance was very good. And this particular survey sensitivity was in excess of 97%, looking only at labs that were querying these particular positions. If we look at lab by lab performance in this heat map, each row is a lab, and blue is good. Blue is that a variant was detected, and then the other colors are some version of either not detecting or possibly not carrying out the test. So material was sent to 111 participants, spread across 3 different specimens. Overall accuracy across all positions was in excess of 98%. Yes, there were some false negatives and some false positives. And we've zoomed in on these in the small insert panel that's on the far right. And if you're interested in seeing a little bit more of this published data, you can pull up the paper that I've given you the reference here. But the key point would be that this survey does include challenging variants such as multi-nucleotide variants with 2 adjacent point mutations or single nucleotide variants. Over time, the CAP proficiency testing offerings have evolved to include more and more of these challenging variants in order to help labs really understand the performance of their assays and pipelines. The other benefit of these proficiency testing surveys is that they do give us the opportunity to ask survey questions about the lab's procedures, including questions that help us understand what platforms are being used for testing, what selection methods or enrichment methods, what kind of assessment is being made of neoplastic cellularity. Those provide a snapshot of current practice, and we try whenever possible to publish those data so that they can be available to the scientific community. So in conclusion, I introduced the idea of performing representative validation when an assay is too big for us to be able to validate on every possible variant, and the idea of methods-based proficiency testing, which is the idea that we will challenge the assay using a representative set of variants that are typical of those that the assay is able to detect. And that's our paradigm for carrying out quality assessment of NGS-based assays. So thank you very much for your attention. As a parting thought, I want to tell you about the Molecular Oncology Topic Center that's available on the website of the College of American Pathologists. And if you were to search for Molecular Oncology Topic Center, you would find it easily. And this Topic Center basically is a repository of interesting peer-reviewed publications and resources that are available from the College of American Pathologists as a service to the molecular oncology testing community. Thank you so much.